Hi, everyone. In this video, we're going to talk about our non-specific immunity. And as I'm recording this, it's also peak of flu season, so it feels very applicable because, yeah, this is something we all need to know how to use. So inside of our body, with our non-specific defense, we're looking at something foreign has gotten in, and we just want to handle it. We don't care what it is. We don't care what strain of virus. We just want to get rid of it. So these are going to be a lot of different strategies for how we can get rid of those pathogens so they don't make us more sick. So looking at our non-specific defense, we're going to split it first up into two major parts. We have our primary or our first line of defense. And so with the first line, it's realistically just don't let it in. So the first line of defense are going to be the boundaries. You can't get sick if it actually never had access to your cells. So these will be things like epithelium, hair, external secretions, so exocrine secretions. And along with this, surprisingly, something that's not even yours is our normal flora. So with our normal flora, what we're talking about here are the bacteria that live within us. And interestingly enough, as you walk around during the day, most of the cells that you're transporting around aren't even you. It's the bacteria that live in you. And they're going to defend their territory. So you have a symbiotic relationship with them. They'll take your byproducts from your digestion, so the things that you didn't utilize, and they'll eat that and live in your digestive gut. So when they talk about gut health, they're talking about those. And if a pathogen comes over there, they're going to destroy it to say, no, this is my territory. And so they'll defend you. When we're talking about exocrine secretions, it's going to be things like the snot and the mucus, tears, sweat. These all will have antibodies in them, which is part of the specific line of defense. So we'll talk about that in a different video. But they're going to defend and attack directly once they come in contact with stuff and then flush it off of the tissues. Hair can keep things off of your skin, so it's further away from ways to get in. But then also epithelium. I think a lot of people think for the first line of defense, the skin. That's a very obvious epithelium. We see it a lot. But it also includes the epithelium inside of your tracts. So the liners for the digestive tract or the respiratory tract. They're going to prevent things from getting in you. Because remember, things aren't in your body until they actually cross an epithelium. So the epithelium is that first line of defense. So now what do we do if it got in? Well, now we're dealing with our second line of defense. And there's going to be a variety of strategies. So let's talk strategy one as cells that eat things. So we'll have our phagocytes. With our phagocytes, we'll have monocytes, which are also called macrophages. The name just changes based on where you're located. So if you're inside of blood, we'll call it a monocyte. But if you're out monitoring the environment, we'll have a macrophage. We have dendritic cells. And we'll also have neutrophils. So these are all the cells that are going to go around and they're going to eat things. They're going to engulf pathogens. And then they'll break them down. Now, the nice thing with these phagocytes is once they find something that's foreign, they eat it, they can show that off to the specific defense. So when you're doing all of these different lines of defense, they're doing them all at the same time and helping each other out. So think of it kind of like a full-grade military attack. And so we don't just want to use just one line of defense. We don't want to use just like the Navy. We're going to go, okay, Navy, Army, Marines, like let's call all of these different defenses in and work together. So speaking of helping the phagocytes, we'll do something called inflammation. Inflammation is going to be triggered by two cells, basophils and mast cells. All right, so both basophils and mast cells, I'm going to group these together, are going to secrete histamine and something called chemotactic factor. Chemotactic factor causes what we call chemotaxis. It's a chemical message that says, hey, come here or go away. So with chemotactic factor, we're going to say either come here. So this is the call in. We'll call that our positive chemotaxis. So you're going to recruit them to come to you. And then you'll also have negative chemotaxis. So positive is the call in. Negative saying, oh, avoid this area. There's a pathogen here. So when we're looking at chemotactic factor, you're causing chemotaxis. Taxis, think of like taxis. You're getting a ride. And chemochemical, the chemical message telling you where to go. And then histamine is going to help make it easier for things to get to the area where there is inflammation or to cause inflammation. So what's happening with inflammation overall is that we're going to make it easier to call in, for example, our phagocytes. If we want to catch these pathogens, we're going to need to call in backup. And we also have to make it easier for that backup to get into the tissue. So histamine is going to do things like increase capillary permeability, All right, so it increases capillary permeability, so it makes it easier for things to get through. It'll also cause vasodilation. So we're going to make the tube larger, and then we're going to put gaps in between the endothelial cells so those phagocytes can squeeze through. It will also activate kinins. And those kinins will do these same things. It's positive reinforcement. So you will also increase capillary permeability. You will also vasodilate. But you're going to have two more things that you're going to do. One of those is stimulate pain receptors. So you're going to activate pain receptors to say, hey, we need to deal with this. There's something in this area that is just not right. And so you're going to call attention to it. But then they're also going to do something what we're going to call CAMs. Our CAMs are our cell adhesion molecules. And so I always think of like inflammation as kind of like dealing with a lazy river. And so you're floating along and then the exit's like right there. And I'm one of those people like, I don't want to get in the water. I like the idea of floating on top of the water. I don't want to like get my hair wet. And so when we get to that exit area, I need the handle to grab a hold of so I can pull myself to the stairs. And that's exactly what the CAMs are. So along our blood vessel, we're going to have these little gaps that we talked about, right? We're increasing that capillary permeability, so we have the little gaps. But remember, these phagocytes are flying by, so we got to put a little handle on their wall. And that's what the CAMs are. The CAMs are little handles, so when we're floating around, we can grab that handle and then swing out into the tissue so that we can have access to the area that needs to be cleaned up. So that's what's happening with inflammation. All right, another one would be immunological surveillance. All right, so with immunological surveillance, what we're doing is we're going to check over our cells. So all of the cells in our body will have a little flag on their surface, a little antigen that says this is where it belongs. So not only is it saying we belong in your body, so all of my cells will have a flag that says belong in the village of Jen, but it'll also say specifically what organ and what it should be up to. So that way, if you catch a cell, like a kidney cell, in a different area, like over in maybe the thoracic cavity, that's an indicator of like, ooh, something's wrong and it shouldn't be here. We need to destroy that. So immunological surveillance will help with things like cancer because it will try to track any of these irregular cells and cells that are in the wrong location. The one that you're going to use to do this job is called an NK cell. The NK stands for a natural killer cell. So the natural killer cells do exactly what their name tells you. They're going to kill things. And the way they do that is by secreting two toxins. One is going to be called granzymes, and the other ones are porphyrins. So porphyrins are going to put pores in. They're going to shoot holes into the pathogen. So think of a bacterial cell, and then it can't regulate its environment, so now it's got holes in itself. So this one's going to shoot holes in it. And then granzyme, I've always struggled with coming with an analogy, and I'll give credit to a student for this one because I was like thinking of grandmas, and he's like, well, if you feed them with a lot of cookies, they'll blow up. And so granzymes make the cell blow up. It triggers apoptosis. All right, our next one is going to be interferons. With a name like that, they're interfering with something. And specifically, they're dealing with viral infections. We will utilize the NK cells again because the NK cells can kill those infected cells. They can also call other cells in the area to say, hey, there's virus here, so we can actually use these more. We will use phagocytes. So phagocytes will also signal out like, oh, we found a virus-infected cell, or we can eat a virus-infected cell. And we will have the viral infected cells themselves releasing interferons. So interferons are basically the chemical message to say, hey, there's a virus inside of our body. Because when a virus comes in, they're going to start replicating inside of our own cells. And so the viral infected cell is also going to warn everybody. So it's kind of like the cell saying, hey guys, I'm making more virus. Sorry, but be ready so you can, maybe defend yourself. So that's what the viral infected cells are going to be doing. They're sending off a signal to everybody to say, yeah, sorry, be ready. All right, so then our next one is going to be our complement system. Complement system, with a name like that, is going to be complementary. It's going to be helping other lines of defense do their job more efficiently. These are going to be plasma proteins, so they're proteins that support other parts of our defense. And so, for example, if we want to help out the phagocytes, we want to make it easier to engulf the pathogen. So if we have a phagocyte coming over, and it's going to try to, so we're going to make them look like a little happy phagocyte, eat some bacteria, we're going to want to make it easier to eat that bacteria, because bacteria know that phagocytes are going to want to eat it. So on the outside of that bacteria, you have a layer that makes it harder to engulf. So it'll squeeze out of its mouth. By the way, it's not actually a mouth. These are pseudopoa that are wrapping around it. So we're going to try to make it easier by adding these complement proteins on the surface. And so the complement proteins, in this case, are called opsinin. And so opsinin is a complement protein that puts handles on the bacteria. And so the analogy I like to use that is if you think of like trying to grab a bar of soap it's going to squeeze out of your hands. So that's what's going to happen when the phagocyte tries to grab it. It's going to squeeze out. And so if we put little toothpicks in our bar of soap, when we go to pick it up, we can hold on to it better. So now we have little handles for the phagocyte to be able to pull on. So that's what's happening here. So this process is going to be called opsonization. So opsonization, which we're going to use the protein opsin for. So I guess we can leave the line down here. All right. Another thing that we can do is the same strategy we saw down here. The granzymes cause a cell to blow up. So we can do cytolysis. That's helpful. We'll do that again. So we'll go cytolysis. We can also help out our specific defense. We haven't talked a lot about the specific defense, but with the specific defense, we can have a pathogen get caught by an antibody. So here's an antibody. This is a protein that is very specific in shape and locks up to a pathogen. So we're going to put our pathogen up here. So let's catch a bacteria, for example. We'll make them look a little evil. So angry bacteria. And we want to get rid of it. And so what we're going to do is we're going to use the complement protein right here. So here's our complement protein. It's going to grab onto the handle part of the antibody, and it's going to help that antibody get a ride on a red blood cell. So here's our red blood cell, and the job of red blood cells is to transport things around. So we're going to grab a hold of our little bus here. So red blood cell bus, a red blood cell. And what we're going to do is we're going to get a ride to specifically the spleen or the liver, an area where you have a lot of phagocytes, so you can then engulf this pathogen. So the antibody caught it. The complement protein right here is going to attach the immune complex. So that's what this is. We have an immune complex here that's going to attach the red blood cell to help move it along. So we're going to call, so I'm ahead of myself. So here's that complement protein. So we're going to call this elimination of immune complex. The other thing that you can do, so if we're kind of keeping track here, how we're helping. So cytolysis, kind of working with like how NK cells do their job. Opsonization, we're helping the phagocytes. Elimination of immune complex, we're going to help antibodies, which are a specific defense. We're also going to reinforce inflammation. It works, it's a strategy, so let's just keep doing that. So we will also cause more inflammation. And so we'll cause inflammation by stimulating the basophils and mast cells to tell them to release more histamine and chemotactic factor. So our job of complement is to reinforce the other strategies that are also going on. Another component for your second line of defense that we can use to reinforce what's happening here is through fever. And to cause fever, we're going to release pyrogens. And what's happening with fever, you ever notice when you have a fever that you feel cold, even though like if somebody comes to touch you, you're like really hot? It's because what you're doing is you're changing the thermostat. You're changing your set point. So what used to feel comfortable for you before now feels cold because it's below what your new set point is. So why do we do it? Well, a couple of reasons. One, our cells are going to be better able to handle that warmer environment than the pathogens or bacteria that are coming in. But two, with an increase in temperature, it makes it easier for molecules to move. And in a lot of these responses, we're moving molecules. So it also helps all these other responses. So if it feels a little messy, that's okay, because you want to have a lot of different variety. And I think one of the things that's a little tricky with nonspecific defense is that a lot of people want to put it in a very linear pathway to say, oh, we're going to do this, and then we're going to try this. But it's more about whatever the bacteria or the pathogen runs into first. We're going to activate it. So if we run into a basophil, we're going to release histamine and chemotactic factor. If we run into a phagocyte, it's going to eat it. But then they'll start communicating to each other and helping each other out so that way you don't get sick with everything else going on. Use all the strategies and then reinforce it so we can stay healthy. Thanks for watching.